Microbiota-derived metabolites regulate neural signaling pathways. How chemicals made by gut microbes quietly influence brain communication. This is the hidden conversation between the gut and the brain. It's easy to think of the brain as separate from the rest of the body, controlling everything from a distance. But there is a constant exchange of signals happening between the gut and the brain. This connection isn't just about digestion or hunger. It involves chemical messages traveling through the bloodstream and nervous system. Some of these messages are produced not by human cells, but by microbes living in the gut. These microbial products, called metabolites, are part of a communication system that quietly shapes how the brain functions. What microbiota-derived metabolites actually are: inside the gut, trillions of bacteria break down food components the body cannot process. In doing so, they produce small molecules known as metabolites. These include short-chain fatty acids, neurotransmitter precursors, and other signaling compounds. Even though they originate in the gut, many of these molecules don't stay there. They can enter circulation and reach distant organs, including the brain. Their role goes beyond digestion, acting as chemical messengers that influence different biological systems. How these metabolites reach the brain: for any substance to affect the brain, it must cross protective barriers or influence signals indirectly. Some metabolites can cross the blood-brain barrier, while others interact with the nervous system through pathways like the vagus nerve. They can also affect immune signaling, which in turn influences brain activity. This means the brain doesn't need direct contact with the gut to be affected. Communication happens through multiple overlapping routes, making the connection complex. Regulating neural signaling at a chemical level, once these metabolites influence the brain, they can affect how neurons communicate. Neural signaling depends on chemical messengers, receptor activity, and electrical patterns. Certain microbial metabolites can enhance or reduce the activity of these signaling pathways. For example, short-chain fatty acids can influence gene expression in brain cells, altering how signals are processed. Other compounds affect neurotransmitter balance, indirectly shaping mood, attention, and behavior. These effects are subtle but continuous. The role of diet in shaping these signals. Because gut microbes depend on what we eat, diet plays a major role in determining which metabolites are produced. Fiber-rich foods tend to support beneficial compounds, while highly processed diets can shift the balance in a different direction. This means food choices influence not only physical health, but also chemical signaling linked to brain function. The connection is indirect but consistent. Over time, small dietary patterns can lead to noticeable changes in how these systems interact. Links to mood and cognitive function. Research has started to explore how microbial metabolites relate to mental states. Some studies suggest connections between gut-derived compounds and mood regulation, stress response, and cognitive processes. While this doesn't mean microbes control thoughts directly, microbes control thoughts directly, they may influence the conditions in which brain signaling occurs. For example, changes in metabolite levels have been associated with differences in anxiety and attention. These findings are still developing, but they point toward a deeper biological link: the involvement of the immune system. Another important part of this interaction is the immune system. Microbial metabolites can affect immune responses, which in turn influence brain function. Inflammation, even at low levels, can alter neural signaling pathways. By modulating immune activity, gut-derived compounds indirectly shape how the brain responds. This adds another layer to the connection, showing that communication between the gut and brain isn't limited to direct signaling. Why this system is still being studied. Despite growing interest, many aspects of this system are not fully understood. The gut contains a wide range of microbes, each producing different metabolites under different conditions. This makes it difficult to isolate specific effects. Human studies are also complex because individual diets, genetics, and environments vary widely. While the general connection between gut microbes and brain signaling is supported, the exact mechanisms are still being explored, what this means for future research and health. Understanding how microbiota-derived metabolites influence neural pathways could change how certain conditions are approached. Instead of focusing only on the brain, future strategies may consider the role of the gut as well. This could include dietary interventions or targeted therapies that modify microbial activity. However, this field requires careful research to avoid oversimplifying the relationship. The goal is not to replace existing knowledge, but to expand it by recognizing additional factors that influence brain function. Final thoughts. The idea that microbes in the gut can influence brain signaling may seem unexpected, but it reflects how interconnected biological systems are. Microbiota-derived metabolites act as messengers, linking digestion, immunity, and neural activity. While much is still being studied, the evidence shows that the brain does not operate in isolation. Paying attention to these connections helps create a more complete understanding of how the body works as a system. Thanks for watching.